Okay, and now for something totally different. This is a story about developing instrumentation that allows you to use the tools of molecular biology in a remote sensing context. So think of it as a way to acquire information about what organisms are present, what they're doing with respect to prevailing environmental conditions without physically being there, without returning a sample to a laboratory for analysis. And it's a project that's been going on for some time, and many people involved, and I just want to point out funding sources: the Center for Microbial Oceanography Research and Education, or CMOR, run out of University of Hawaii, Dave Carl et al., folks at NASA Astrobiology, and the Moore Foundation have had a big input into today's work. The first image that I saw that kind of captures this concept of an instrument that applies molecular biology in the sea is shown here. And it came out of the University of Washington, Seattle. And I believe Ginger Armbrust had a big hand in creating this. But the idea was really pretty simple conceptually. There is some device, and it is sort of taking in all manner of organisms and out a sort of metaphorical array of data that streams from it. And what's important about this concept is that it's an instrument that occurs in the context of an ocean observing system. It's not just there by itself. There's a lot of contextual data. It's very rich. And our vision of what this group termed ecogenomic sensor is not only one that is static, it's immobilized to the seafloor or something, but one that's mobile and capable of moving with coherent populations. So with that, I'd like to just give you a sense of what I'll touch on. What is an ecogenomic sensor? I don't think we've got a definition for that. I'm going to offer a kind of prototypical version of that, the ESP, give you a sense of what it looks like and how it operates, give you some idea of applications, and I want to plant a seed for what we might do in the future with these types of devices. So first, to the definition. I define an ecogenomic sensor as a device that allows for application of molecular analytical techniques outside of the lab. It's completely hands-off. It acquires the sample, processes it, it transmits data, and importantly, it allows for two-way communication. That is, it not only does something and just sends data, but it allows you to go back to that instrument and retask it, to have it do other things, to alter its mode of operation. And that's very different from what a lot of oceanographers practice, which I'll call ecogenomic sensing, where they apply molecular techniques, sometimes outside of a laboratory, but they usually have to bring their lab with them, or parts of it. And they're there for a minimal amount of time. And generally, there's a human in the loop. At some point, a person has got to get in there and manipulate samples in order to get them processed. So ecogenomic sensors are totally hands-off. Here's an example of this prototypical device that we call the ESP, for environmental sample processor. It consists of a core, which you can kind of see in this photo, about 1/2 meter by 1 meter. Think of it as a double-wide kitchen garbage can. It's about that volume. We call it lab in a can. The core allows you to collect material, do sample archivals, some real-time probe arrays, and I'll give you some examples of that. It also supports things that are outside of the core, things that allow you to do something special with the samples, work in the deep ocean, or maybe acquire sort material so that you can enrich for certain targets. But they're devices that live outside of this fundamental core. And at the back end, we have what we call analytical modules. Think of these as point-of-care diagnostics. Lab on a chip is a great example. Those are wonderful devices, but they require a lot of upstream sample handling, processing before they can actually be used, particularly for environmental samples when you may need to work with liter quantities or more of material and go through and sieve it down. So those are the main parts, and I'm going to focus primarily on the core and an example of an analytical module which uses quantitative PCR just to sort of illustrate this system. So here are the current functions. We use it to apply DNA and protein arrays. And this is a very simple technology, but the point is you get a sample, we homogenize it, and we can use the crude homogenate directly to capture molecules of interest using antibody or DNA probe technology. You can capture an image of that array and broadcast it. Quantitative PCR. Uses the same homogenate that we can, if you like, from the DNA proberase. Does a solid phase extraction. Applies a sequence of quantitative PCR reactions in series, and repeats. And last but not least is sample archival. And I'm not going to talk about this today, other than to say that I think this is one of these fundamental things. We always want to be able to bring material home. Interesting things always happen on nights and weekends when you're not there, holidays. You'd like a way to verify the data you see in real time is actually real, and you'd like a way to dig deeper with conventional laboratory methods that just aren't appropriate to think about putting under the ocean and running on 12 volts. And I want to mention work that's being done by Ed DeLong and his colleagues at MIT, showing that we can get very good preservation of messenger RNA for over a month at ambient temperatures, room temperature. So it's a way to collect material, preserve it, no flash freezing, and get good messenger RNA out 30 days plus later. And we've also shown that this is good for proteins as well. So if you're interested in that, I'd be happy to talk to you about it. Okay, so here's a brief look at this chemistry for the ribotyping arrays. We have a filter we print probes on using a piezo printer, inkjet printer. Capture ribosomal RNA, signal it, it's off-the-shelf chemistry, get arrays. Turns out to be a very highly quantitative type of assay, but this was the most important. It's why I started with this assay. It's direct capture of sequences from crude homogenates, no purification. Reagents are very stable for months at room temperature, all liquid. With respect to PCR, we have this separate device, kind of an analytical module, if you will. This is about 18 inches high. It just shows this layout of all the equipment that actually operates this. The key pieces here is a reusable solid phase extraction module, PCR module, which is shown here. That's a quarter for size, so it's a little bigger than your fist. Comes out of Lawrence Livermore Labs, out of the hunt for bioterrorism agents, actually. And then our PCR reagents are stored in tubing coils, and we've gotten up to 5 months stability of quantitative PCR, real-time PCR reagents, without any degradation at room temperature. Here's how the microfluidic block works. We start with a sample which has gone to our extraction module. We have PCR primers and probe enzyme up in those coils. That extract, once we have purified DNA, moves to a valve port. We take a sample of our selected primer probe combination, mix that with the slug, with the sample. It runs into the module, thermocycles, acquire the data, and then that could actually go off to some other analyses. Right now, we simply throw it to waste, and we move on. So here's how they work in concert with one another. Here are two examples of these arrays. That's about maybe half the size of your thumb. Number of probes are printed. This is not as high density as we can get. It's just to illustrate a bunch of different ribotypes of some pretty garden variety marine bacteria. And the names are not important. What's important, just sample 1 and sample 2, these are environmental samples, been developed, these arrays have been developed on a mooring at sea, on batteries. And if you just notice from time 1 to time 2, there's a difference here in the abundance of ribosomal RNA here to here. And it happens to be group 1 archaea. The same homogenate that was used in these arrays is parsed and run for quantitative PCR. There's a variety of runs, but here is very abundant in that sample. And in time 2, sample 2, you see that shift. We've lost the transcript, the ability to see the structural RNA, but we can still see the gene. So that's just an example of how we're using these two things. And that takes about an hour or 2 hours or so to develop an array, a few hours for the PCR to come off. Here's the platforms that we've used to deploy this machine, a whole bunch of different things. Moorings for coastal work, remotely operated vehicles, that little thing sticking out as a wand so you can sample with a mechanical arm. An elevator, a free fall vehicle, you throw this off, it sinks to the bottom and move it around or leave it where it sits. A drifter, which I won't have time to talk about, or piers, where it's just simply sitting there collecting water on occasion. This drifter is very interesting, and I want to just pause a moment and think about that. Most of the molecular analytical work we do in the ocean, we collect a sample and we reference it immediately to the prevailing environmental conditions at that moment. But the biology is a product of the past as much as it is of the present. And so the notion behind this drifter is to follow coherent populations so that we can put that gene expression into the context of their history, not just that instant in time that you sampled. So as I said, it's a whole different story, but if you're interested, I'd love to talk to you about that. Okay, here's an end member of how we envision using this device. Most fundamental is in water quality monitoring, a device that's immobilized, or a series of them. It takes sample, gets data, registers it, and look on your iPhone, don't go swimming. This is a much more challenging case, open ocean, microbial mediated biogeochemical cycles, and this is something that's really being driven by the Seymour Science and Technology Center and John Zera's group at UC Santa Cruz. So here we have an array of devices that are looking at these various pathways for elemental cycling, biogeochemical cycling. I'm going to start with that first case of kind of, looking for that one needle in a haystack that tells you something that's nasty going on. And it's a case where we went and deployed instruments down here in Southern California. And we have two of these devices that are moored off Newport Beach. They have names, they're network computers, so they have names, Mac and Bruce. So Mac and Bruce are out here, and they're immobilized at about 15 or 20 meters or so below the ocean surface. And the idea was that harmful algal blooms of this organism, these are pennate diatoms of the genus Pseudonychia, actually start in the subsurface. And they are brought to the shore through a physical transport mechanism. So when people are searching for them, usually they're taking samples from piers or nearby, and you don't see these bugs, and then they suddenly just appear. And the idea is, how do they get there? They make this little molecule, which is called domoic acid. It's an analog of glutamate. It's a neuroexcitatory amino acid, and it is harmful to humans and wildlife. So our objective was use the ESP subsurface to look for the bugs and the toxin, and see if we could predict an event, a harmful event, before it actually happened. So here we are. The instruments are deployed. We used a cell phone modem on shore to get a radio signal from the instrument to the cell. From there, we have an internet connection. That was back to us here in MBARI in Monterey Bay. Now, the whole idea was to run this autonomously, so we're not present, we're not on site, we would just operate the instruments. But unfortunately, I was in Death Valley with my family, and I had no way to connect with it. I happened to notice that some of our neighbors around us had small dishes, and they were obviously on the net. So I looked around, and lo and behold, there's an unsecured wireless network, and I hopped on. And so now I'm in Death Valley running instruments that are doing DNA probe analysis for not only specific organisms, but also toxins. And I'm in the desert. It's remarkable. Here's just some example data from these two machines. And what you're seeing on the top in this kind of purpley color is the depth, the tide. These are anchored to the bottom, so as tide comes in and out, you see this tidal signature. And this is temperature, and what we were looking for is exactly this big dip. You see these two dips. And that's cold water, as water's being brought up from the depth and moved into shore. So here is an example of one of our arrays. It's a small snippet with a variety of these nasty bugs that the ribosomal RNA probes for. Here's that sequence. And here's just some raw array results. And what you'll see at these various time points, it's pretty obvious that suddenly, over this period, we just start to hit big on these bugs. They're present. And the toxin goes from non-detectable to something which is not tremendously hot, but it's there. And this is important because these organisms, the toxicity is something that's physiologically regulated. People don't really understand how that works in nature. But point is, we're able to say, yeah, they're there and they're toxic, but they're not super toxic. And that was done, again, from anywhere you had a wireless connection, you could watch this happen in real time. I'm going to switch now to some of these microbial community dynamics, which I think are far more challenging questions and much more interesting in a lot of ways to me personally. You often are, you're deluged with a lot of data, and no one can ever read this, and so I just point out that we're looking at how this instrument is working with respect to atmospheric wind forcing. You look for chlorophyll, all sort of optical properties, physics, chemistry of the water, look at quantitative PCR for what genes are present, what ribosomal RNA community is present, and this is embedded within this oceanographic context. Now, just an example of some of the data from that, we keep picking on our friends, the Krens, but you look at how abundant are they with respect to observed concentrations of nitrate, salinity, and temperature, and it's exactly what you might expect. These organisms are often in the ocean or in the deeper waters. They're very abundant in high nutrient, low temperature, high salty water. Their abundance declines as you get in lower salinities, higher or lower nutrients, and warmer temperatures. So it's exactly what you'd expect. But if you keep running this over and over again, and this is something that Julie Robidard and John Zer's lab has done, here's that expected, what we would think we would see, the abundance of these in the springtime being driven by this upwelled water that's pushing those organisms, the same mechanism to deliver those HAB species in Southern California. You expect them to be there, and they're there. But what Julie's noticed is that they're also there really abundant in the fall. And what's important about this is that these bugs are also nitrifiers. And so we believe that they are playing a pretty important role in the nitrogen cycle within the bay to help fuel these algal blooms that become very prominent in Monterey Bay in the late fall when upwelling is suppressed. We don't have a lot of upwelling. And that's something that Julie's following up on. Okay, I'm going to move to the last example, which is now to take this technology to the deep ocean. And what we've done, this is really funded by NASA Astrobiology. Someone pointed out that this thing looks like an early atomic bomb. It's not. It is our instrument, the same one that we've used on the pier or in a mooring, but it's in a sphere that allows it to go to 4,000 meters depth. And we're going to deploy it in a couple places, but most importantly out here in Santa Monica Basin, where are these bizarre methane seeps. And it's hard to see in this photo, but here's our instrument perched on this mound, which is about 10 meters high off the seafloor, about 100 meters in diameter. It's like a blister on the seafloor. And it has methane that kind of shoots out of it. Sort of occasionally there's little bubbles. Every now and then you get a jet. It's like, just think Yellowstone. There's some guys are shooting off of methane gas at about 800 or 900 meters depth. And I won't get into this. I just wanted to give you an example. You can't see it at all, but here's the instrument. And the point is, when we're in Santa Monica, we can see genes highly enriched for the particulate methane monooxygenase and the ribotypes that go with that. If you move off to point conception, you lose that enrichment of methane. You lose those bugs. They drop off. You see changes in the ribosomal RNA community profile. So the point is, we're really at a point where we can begin to think about running this at all kinds of bizarre places where you can't be for extended periods of time. And we want to look at the microbial community dynamics as a function of how the chemistry is changing, and also in event detection. So if there is a burst of something, that's when you want to collect samples. We're also looking at this as a way to do in situ incubations. And so we heard the opening presentation. There was talk about doing the problems of collecting organisms and water from the depth and doing in situ incubation. So this is exactly the kind of thing we envision doing, being able to test metabolic activity autonomously and interactively, and push that up to a year of deployment duration. Okay, so I just would end here to say that, if you think, where was genomics 20 years ago? I mean, it was, a number of us were laughing. Dan, in fact, our previous speaker, and I were sort of joking. We overlapped for a while at Woods Hole, and we used to pour acrylamide gels, and Dan cut his own combs to get these really tight wells, And that was a far cry from where we are today. And I think that's a good metaphor for where we are today with this class of sensor. I mean, it's sort of like standing there asking Orville and Wilbur, saying, yeah, that's cool, but when do I get transcontinental flight with personal entertainment, And it's coming. I think we've shown that this concept is valid. We can put these sort of things in the ocean. We can run them for pretty extended periods of time. We certainly, with the advent of these ocean observatories that are coming, it's creating a real ripe opportunity to start applying this genomic information in this remote context. It gives us a backbone to hook up on. We don't have to do it all. And I think the way I look at this is it's for the first time we can start to think about how you test a hypothesis, but do it remotely, like we were doing with those HABs. We know what we were looking for. The question was, I want to sample before and after this water mass comes along. And we were able to do that with a wireless connection, even though the instrument was some distance away. So this is something new. This is what we're working on right now, is to take that same functionality and really compress it into a small space and put it on a mobile platform. This is a long-range AUV. It's meant to be out for about a month at a time. It has communications, adaptive sampling. It can kind of hang with water masses and drift, profile. It's a pretty remarkable vehicle. And this is our vision for how our instrument here, lab in a can compress into that space. But you can also do the same thing with profiling floats. So we sort of think of the future as coming out on a ship, seeding the ocean with a variety of these devices, and using the ship as a hub from which we manage these assets. Okay, well that's it. I hope I've helped catch us up on time a little bit. A lot of people involved. I'd be happy to take any questions you have, or if you want to see what a methane plume looks like in Santa Monica Basin, I have it on my laptop. Thanks.
Thank you for a great talk, Chris. This type of studies are literally gold mines for metadata, which is something that we badly need for comparative analysis across various microbial communities. So, any questions?
How many arrays do you try to fit in one of these cans, and do you try to reuse them, and what's the constraint on that?
No, we're not reusing them right now. We run it very conservatively. We'd run maybe 40 of those arrays, or 44, 45. Yeah. And they're printed on nitrocellulose filters, so we're only using them once. There's other ways to skin that, but we're just run very conservatively. Other questions? Yeah.
Is there a sort of open source functionality so that we could develop our own modules for these?
Absolutely.
devices?
Yeah, and thank you for asking that question. Yeah, what we've tried to do is to establish a standard on the back-end analytical module, RS-232 communications, 10 to 14 volts, or less you want to run in your own power supply. And then we have a standard fluidic connection that we use. So that's exactly how we put that PCR module in there.
Could you miniaturize it so it could be swallowed by a person?
With enough time and money, you bet. Yeah.
I have an equally crazy question with that. Have you thought about applying it to studying possible alterations of microbial communities with earthquakes and possible modifications of the microbial community before or after earthquakes? We know that there is a shift in the magnetic field before and after the earthquake. So,
I personally haven't thought about earthquakes. It's interesting.
Well, I didn't think about it until now as well.
But we have talked with people who do water remediation, wells, for example, where you set up some remediary treatment and you need to look over time, and these guys are going out in their van and collecting samples, bring it back to the lab. They would operate that way. I think the best would be, in places that are remote, that requires some time-varying view of what happens. That's where this application is, for this device, would be best applied.
Excuse me, Chris, how hard it is to save PCR amplicons rather than throwing them to waste?
It's not that difficult.
Because archiving makes a lot of sense in this respect as well.
Yeah, you could save your amplicons. Yeah, absolutely.
How much does it cost, and how soon will it be economical for regulatory agencies to have one, or a couple?
The device that I showed you here is commercially available. Just this past year, I think they cost, they're approaching maybe $200,000. They're not cheap. We're building, I have no ownership stake in this, by the way, I should, just as a disclaimer. The company that's building in McLean, they're building the first cohort. There's about 10 instruments that have been ordered, people using government labs and others. I think we're probably a few years before, or maybe 5 years before we get the really small version that would be very purpose-built for regulatory agencies. You know, they don't want the, it's like the custom race car as opposed to the Toyota Prius, right? You know, we've got a race car right now. We've built it custom to do a lot of things. But for regulatory agencies, they want it to be do one thing, do it really well. And so, you know, that's probably a few years away. And I think the cost is going to come down dramatically. You know, I would imagine, like, just think about quantitative PCR. It was over $100,000 for those first machines, and now you buy a much more capable, tiny machine for, you know, 25% of that. So it's the same trajectory. Other questions? Lunch? Music? All right. Hey, thank you very much. I appreciate it.